It's a great pleasure to introduce Professor Alain Fischer. His work is concerned with the diagnosis and therapy of human immune deficiency disorders. His work has led to revolutionary advances in diagnosis and therapy, particularly in the field of gene therapy for these devastating diseases. Professor Fischer trained in medicine and biochemistry. He completed a residency in pediatrics in 1979, and after a 2-year stint at University College London, he joined the Hospital Necker in Paris, eventually becoming the director of the pediatric immunology department. He also served as the leader of several INSERM research units, as the founding director of the Research Institute of Genetic Diseases, Imagine, and was appointed to the chair of experimental medicine at the Collège de France. Not surprisingly, he has received many honors, including the Ernst Jung-Preis für Medizin in 1998, the Louis-Jean Prize in 2001, the Novartis Prize for Clinical Immunology in 2001, the Robert Koch Prize in 2014, the Japan Prize in 2015, and he is also a member of many learned societies, including the French Academies of Science and Medicine, the EMBO Society, and the US National Academy of Sciences.
Thank you very much for the introduction. And first, I'd like to mention that I feel very honored and happy to be part of this Jung-Symposium today. I mean, this is quite a substantial recognition, and thanks for that to the Jung-Stiftung. So indeed, over the last 40 years, I've been interested in inherited disorders affecting the function of the immune system. And I will develop the work we have done on one of them, named SCID-X1. As you will see, SCID-X1 stands for Severe Combined Immunodeficiency X, because the gene is located on a chromosome number, on the chromosome X, and 1 because this was the first described. But first of all, I'd like to mention at least some of the people with whom I've been working over these years. You can see their figures. They look happy. I hope they are still happy to have been or to are still working with me, coming from different places. Some of them have been especially involved in the work I'm going to discuss now, like on the first row, Marina Cavazzana, Geneviève de Saint-Basile, and then on the second one, Salima Hassanbeh Amina, among many others, as you can see. Okay, so over the years, many inborn errors of immunity have now been unraveled. Excuse me. So diseases caused by mutations in a single gene. You can see as a function of time, today there are something like 450 diseases that have been described and associated with mutations of a given gene. This is critical for two things. For medicine, because each time one understands at least the genetic basis of a disease, we can offer a kind of genetic diagnosis, including genetic counseling. But also, this is providing us with key informations about how the immune system works or doesn't work in these cases, in terms of controlling infections, controlling autoimmune diseases, or even controlling cancer. So I would like to focus on a group of diseases named SCID. I mentioned that SCID stands for Severe Combined Immunodeficiencies. These are a group of rare diseases, 1 in 50,000 births, where the characteristic is that T lymphocytes are absent. And T lymphocytes are key cells, mediators of so-called adaptive immune responses that we need in order to fight all of the pathogens with which we have to live. So on this scheme, which is a simplified view of lymphocyte differentiation, starting from the left-hand side with hematopoietic stem cells to the right-hand side with NK lymphocytes, CD4 and CD8 T lymphocytes, and B lymphocytes. So each time you see a vertical bar means that there is an interruption, an abrogation of cell development caused by a genetic disease. There are several groups here described in purple. I'm not going to review them, just let you know that their studies have been quite interesting and I would say instrumental in understanding better how lymphocytes do develop in humans and in general. And I will focus on a group of these diseases, those related, as you will see, to the common gamma chain cytokine receptor. You will see in a second exactly what it is. And one of these diseases, actually this is the less rare among these 15 or 16 SCID diseases, is the one, SCID X1, caused by mutations in the gene encoding this so-called common gamma chain. So let's talk a little bit about this common gamma chain. So what I will review from now is what is the gene that is mutated in that disease, to the understanding of the pathophysiology, and beyond pathophysiology, the physiology related to this cytokine receptor subunit, to the treatment by gene therapy, and a few more informations we collected into this work. So the common gamma chain that is deficient in this severe combined immunodeficiency is part, as you can see on this picture, of the receptor for interleukin-2. Interleukin-2 is a very important cytokine for T-cell function. It has 3 receptor subunits, this common gamma chain, but also the alpha and the beta. At the time the gene was identified, it came as a surprise because IL-2 is not supposed to be involved in T lymphocyte development, and I showed you on the previous slide that T lymphocytes are absent in such patients, plus NK cells that are also absent. But this discovery led, actually made by several people, including the lab of Warren Leonard in the US, to understand that this gamma C, or common gamma chain, is actually part of several cytokine receptors, not only the one of IL-2, shown on the left-hand side, but also the receptor for interleukin-4, interleukin-7, interleukin-9, interleukin-15, and interleukin-21, all of these cytokines playing important roles in lymphocytes function. But based on these findings, it was understood that actually interleukin-7, so the third one starting from the left, is the one that is key for early steps, as you will see in a second, of T lymphocytes development in the thymus, and that interleukin-15, so the fifth, starting from the left, is the one that is critical for early steps in NK cell development. So with time, over the years, the 90s, we understood better the pathophysiology of SCID X1 and the related deficiency in the kinase JAK3 that is associated with the common gamma chain downstream to signal when the cytokine binds the receptor. We did some studies. Jim DiSanto in the lab made the first knockout mouse for gamma C, or interleukin-2 receptor gamma gene, as it is shown. And he showed that actually in the absence of expression in the mouse of the common gamma chain, there is, as it is the case in humans, a block in T-cell development. But he could determine that this block occurs very early, among the very first step of lymphocyte development in the thymus, prior to the stage, for instance, of the rearrangements of the T-cell receptor, which is a key step of T lymphocyte. So this is where there is this small blue rectangle. This is at that stage, the very first stage of intrathymic T lymphocyte development, where the common gamma chain associated with the alpha chain of the interleukin-7 receptor triggers, upon binding of interleukin-7, survival and expansion signals of these lymphocytes. So with time, we really were able to understand the physiology and the pathophysiology of this severe combined immunodeficiency. But by doing so, Geneviève de Saint-Basile, another scientist in the lab, made a very intriguing observation. So a patient was referred to us from Germany, actually, who had an atypical X-linked severe combined immunodeficiency. This child was age 6 years when we investigated him. He had a mild immunodeficiency with some infections, but this was really mild, in contrast to the fact that his maternal uncle died at the age of 6 months, and he died from a severe combined immunodeficiency that is with a complete absence of T lymphocytes. But this child had some T lymphocytes, actually approximately half of the normal count, but they were all memory, suggesting that there were no more any production of naive T lymphocytes. He had no NK cells, and he has some antibody deficiency. And what Geneviève showed, that actually this child had inherited from his mother the very same mutation that led to the death of his uncle, but that very surprisingly, the mutation was found from the left to the right in neutrophils, the PN, in B lymphocytes, the CD19 positive cells, in monocytes, the CD14 positive cells. So there is in the triplet, which is in wild type TGT is TGC. But surprisingly enough, in his T lymphocytes, so the fourth panel from the left, the CD3 positive cells, there was a wild type, the normal sequence. So this observation, as strange as it looks, actually could only be the consequence of a reversion of the mutation, so a somatic genetic rescue that led to the development of some lymphocytes. I will precise what I mean by some. So indicating that this event, as rare as it was, determined a strong selective advantage to lymphocyte progenitors. And actually, such events of somatic genetic rescues have been now described in many genetic diseases of the hematopoiesis, including in 14 skin diseases, indicating that as rare as they are not so uncommon, and that their selective advantage may provide some changes in phenotypes. One question is when this event occurs. So here you have a scheme of hematopoiesis, starting again from stem cells on the left-hand side, up to the many different cells in the blood. So in green are indicated a potential origin of reversion, that is that maybe a T lymphocyte precursor, a pre-T cell, had this reversion leading to the development of T cells that are long-lived, whereas there is no other cells in the blood, neutrophils and so on, in which this reversion is detected. But an alternative hypothesis is that the event occurred much earlier during hematopoiesis, maybe even earlier during hematopoiesis in embryogenesis, so that there were many cells generated from this reversion, but the consequences at the end is that because there might be no selective advantage, or only a transient selective advantage provided by the expression of a normal gamma chain, only in T cells would be the detection of these events occurred. And be detected, not occurred, excuse me, I made a mistake, being detected. So we do not know, perhaps both of these hypotheses can be in place in different cases, but this is not that uncommon to observe such events. So we went further at that time to determine how diverse were the T cells found in the blood of this child, generated, let's admit this hypothesis, by a single cell, the one labeled with the orange rectangle here, a single T cell precursor. So we looked at the diversity of the beta chain of the T cell receptor, so as an indicator of diversity, and we found approximately 1,000 different sequences of these T cell receptor V beta chain in T cells from the blood, accounting for approximately 1% of diversity of memory T cells, since I remind you that this patient had only memory T cells. So it can be easily calculated that to go from 1 to at least 1,000, there has been in the thymus 11 division cycles prior to the stage at which T cell receptor rearrangements occurs. And this is certainly an underestimation, but it shows how potent is the selective advantage provided by this reversion. So we considered that this partial correction of the disease was a kind of natural gene therapy that provided this child with some correction, not a full correction, but some correction of his immunodeficiency. So the conclusion of this part of the work is to say that a very limited set, perhaps only one T lymphocyte progenitor, is able to generate a long-lasting and quite diversified T cell repertoire with efficacy in fighting infections. Then it also suggests that T cell progenitors may be relatively long-lived, have some self-renewal capacity, and this was when we observed that the basis for developing gene therapy of that disease, and with some success, as you will see now. So Salima Hassanbey Abina in the lab developed a vector based on what we knew at that time. This is the late 90s. We were using so-called retroviral viruses, excuse me, originating from the mouse. These are viruses that can infect hematopoietic stem cells and induce, as a retrovirus, their RNA to be retrotranscribed into DNA, and the DNA to be integrated into the genome of cells. So the figure of the genome of the virus is very simple. It includes the cDNA for IL-2RG, so including the common gamma chain. And we were using an enhancer activity located at the extremities of this material, the so-called long terminal repeat, to drive the expression of that gene. So a number of studies were performed in vitro and in vivo. I'm not going to discuss them, but just to tell you that they were positive and led to the development of a first clinical trial that was performed starting in 1999. So the principle, as shown on the left scheme, is that by harvesting bone marrow from a patient, this is a cartoon from one of the patients who was treated, selecting ex vivo the early precursors in the bone marrow by the expression of the CD34 molecule at the surface, then to culture these cells with cytokines in order to induce these cells to divide. This is important for efficacy of treatment. And then 3 cycles of infection with the virus I just mentioned, the retrovirus vector that is unable to replicate by itself, but is able to infect cells. And once this was done, the cells were reinjected to the patients. So what is occurring at the cellular level is shown on the right-hand side. This is the principle. You see the vector, the virus, including the cDNA for gamma C. So upon binding and entry into the cell, then the RNA from the virus is retrotranscribed into DNA. These provirus can be in contact in cycling cells with the genome, with chromosomes, and events of integration can occur, as depicted here. That occurs in a semi-random manner. I will come back in a second to that. But once the gene is there, it is replicated each time the cell divides, which is the case with cells of the hematopoietic system. They can be transcribed, this gene, under the control of the LTR, as I mentioned. And then, of course, the mRNA may be translated, the protein expressed, and at the surface and inducing a function as expected. So we initiated a study in 1999, and soon after, 2 years later, a group in London of Adrian Strascher performed a similar study, and here is the overall result of these 20 patients treated first in Paris, then in London, for SCIDX1 by this technology. And up to now, 18 of them are alive, and more importantly, 17 of them are disease-free. That is, they have an immune system and they can cope with infections. You can see that the median follow-up of these patients now extends above 20 years, up to 23 years, and so these are now young adults living normally. They have a correction of their T-cell mediated immune functions. Their quality of life is normal. Still, a majority of them requires immunoglobulin treatment because the B lymphocytes also need this common gamma chain, and with the technology used at that time, there was no correction of the B lymphocytes. I will come back to that limitation of the study. This is what we call the vector copy number. That is the number of the transgene found in peripheral blood mononuclear cells, so the circulating cells. So each color line curve indicates one given patient, the one treated in Paris, and you see that they are detected over time with actually 1 copy per T lymphocyte, accounting for an overall number of 0.6, 0.7 per peripheral blood mononuclear cells. These are the T lymphocytes, so they were at 0, of course, before treatment. And after some initial variability, you see that upon time, the T lymphocyte counts are within the normal values marked by the dotted lines, or slightly below or slightly above. So it looks pretty good. But unlike what we observe with T lymphocytes, this is not the case with other cells. NK cells are still absent, as you will see. And no B cells, no monocytes, no neutrophils being transduced were detected. So only T cells were corrected, and one can detect the different T cell subsets. On the left-hand side, the CD4 T cells with the naive, so the cells that have not yet been activated by an antigen in vivo, and on the right-hand side, the CD8 and naive CD8. So there is a trend to see a decline in naive cells, but still they are detectable more than 20 years after treatment. Another way to detect newly formed cells are the so-called quantification of new thymic immigrants that express a piece of circular DNA, the T-cell receptor excision circle TREC, that is the product of rearrangements of the T-cell receptor in the thymus. You see that these TREC are still detectable after a long time after gene therapy, indicating that there is a sustained detection of such cells. So this was quite a reasonable, successful result. Nevertheless, it was also associated with some toxicity because the enhancer shown on the right-hand side of the vector here could induce transactivation of other genes of the genome, and in some of the patients this led to malignancy that was efficaciously treated, but still was a problem related to gene therapy. So to solve that problem, together with many collaborators worldwide, we designed so-called self-inactivating, sorry, there is a mistake, it should be self-inactivating vectors, where this enhancer has been deleted, and in order to get expression of the IL-2RG gene, a promoter has been added, which is the one of the short form of the elongation factor 1 alpha. So another trial was performed between Paris, London, and several centers in the US, and it led actually to a reasonable success, that is clinical benefit, no toxicity, there was the death, but unrelated to gene therapy. These are on the left-hand side, the T lymphocyte counts, on the right-hand side, the copy vector in the T lymphocytes of these patients. So indicating that this trial was as successful as the first one, but now was safe and no longer at risk, apparently, up to now, with a follow-up of more than 10 years of the complications of the gene therapy. I already mentioned one result that is surprising in many ways, that is this technology led, as I showed you, to reconstitution of the T lymphocytes pool with a clear-cut clinical benefit, but NK lymphocytes detected early after gene therapy, as you can see on the left-hand side part of this curve, are no longer detectable more than 5 years after gene therapy, marked by the arrow. So we can conclude that obviously, this is the least we can say, that the correction of the NK cell deficiency is much less important than the one for the T cell immunodeficiency, and this may be related to a reduced production of NK cells from the precursors and or a shorter lifespan. And the second conclusion, which is a bit more surprising and a little bit provocating, is to say that there are limited clinical consequences, since these patients are no longer patients, actually, they are doing well, suggesting that at least for some time there is a redundancy of NK cell activity in this context. So another aspect of these studies we were interested in is to really understand better what was going on in this gene therapy of SCIDX1 in these trials. So prior to gene therapy, this is a very simplified scheme of the hematopoiesis in these patients. From stem cells, hematopoietic stem cells, they were able to generate all of the myeloid lineages, neutrophils, monocytes, and so on, as well as B lymphocytes, but no NK, no T lymphocytes. Once they have been treated, we still have their unmodified hematopoiesis, plus, in addition, transduced modified T lymphocytes now that were generated by the gene transfer. So we have this combined situation, which is a little bit unorthodox, because it suggests that there has been, at least for 15, 20 years, the persistence of precursor T cells able to generate T lymphocytes. We were also interested to understand how many of these transduced progenitor cells were detected in vivo and gave rise to the T lymphocytes. So the question is whether it's only a few or millions of such cells being corrected ex vivo and generating T cells, as shown in this very simplified cartoon. So to do so, it's possible to look at, by sequencing the genome of the T lymphocytes from these patients, to look exactly where the retrovirus has landed into the genome, and to calculate and count the number of integration sites. And so we have the sequence, so we know exactly where they are. So by doing so, and you have the results for some of the patients here, you can see that the number is quite low. It's around 500, 1,000, something like that. But even if there is some underestimation because of the sensitivity threshold of the method, it tells us that the answer to the question how many transduced progenitor cells there were is at maximum 1,000. So telling us that so few cells were able to generate a full T cell pool in these patients. So indicating kind of auto-amplification of the gene therapy. And this is stable over time, which is, of course, good news for the patients. Another question is, of course, and very important, is to address the one whether these T cells that were generated in normal counts, are they polyclonal? So do they have a diverse T cell repertoire? So that are they efficient to recognize the many antigens from different pathogens? To do so, excuse me, we sequence the T cell receptor beta of these T cells, the T cell receptor beta gene of these T cells, and the results are shown here on this slide. You have first on the upper panel of the left-hand side the results in controls, adults and children, and you have to read what the blue part of these lines, indicating, for instance, that there are approximately 20,000 different T cell receptor beta chains detected in controls in patients as well as in adults. And then the other figures from the lower part of the left panel, as well as all of the right panel, show the data from patients treated by gene transfer. And you see this, with a single exception, that the number of T cell receptor beta sequences is similar to the one of controls, indicating that the gene therapy has led to a full diversification of T cell repertoire. And one can calculate by knowing the number of transduced clones I mentioned earlier and the diversity of this repertoire. And this is shown here over time, that the minimum number of divisions that occurred in the thymus from the modified cells up to, for the generation of the T cells that are detected in the periphery, is something like 10, or sometimes a little bit more. So showing that there has been an expansion of T cell precursors prior to the rearrangements of the T cell receptor beta chain. So this is figured here. This maybe should, it looks busy, but here we start from the left with 2 of these clones, so among the 500 or sometimes only 50 in one patient, that were transduced ex vivo. So now they can express the common gamma chain. This is the orange mark. And then in vivo they can expand. Here a few cell division cycles are shown, but of course many more, and actually more than 10, generating T cell, precursor T cells that went to rearrangements of the T cell receptor, so looking inducing their diversity. And in the periphery, these are the cells on the right-hand side with a different abundance, but this is very much diverse. So here the point is to reiterate that there has been a massive expansion of transduced T cell precursors, at least 10 times and probably more in the thymus, explaining the success of this gene therapy, despite the fact that the number of corrected cells is limited. So today, where do we stand? We had the first step of gene therapy, I mentioned, where we found efficacy but also some toxicity. So this led to an evolution where we were using these safer vectors, the so-called self-inactivating gamma retroviruses. And now there is a third step with further advances, where instead of using gamma retroviruses, one is using, as for other gene therapy trials, lentiviral vectors, still a retrovirus, but able to correct more cells, and especially stem cells that are non-dividing. And in addition, by adding a little bit of myeloablation, one can get rid of some non-transduced stem cells in the bone marrow and replace them by transduced cells. And this is leading actually not only now with the correction of the T lymphocytes, but also of NK lymphocytes and B lymphocytes, so that these patients do not require any more immunoglobulin therapy. So the conclusions here are to say that from these trials, we observed a sustained correction of the T cell deficiency, and that the safety issue I mentioned has probably been fixed. And now we go to an extension I will briefly mention to the treatment of other diseases, especially other primary immunodeficiency. We then learn more about the fact that T cells are long-lived and that a very small number of precursors can generate many cells, and then finally that there are self-renewing T cell precursors. So from there, over the last years, a number of diseases of the immune system are now targeted for gene therapy. There is a list on this table. I will not go into the detail, just to tell you, you can see that approximately 10 diseases that have been treated. I will briefly mention one of them, which is the so-called Wiskott-Aldrich syndrome. This is a more complicated disease where there is not only a problem with lymphocytes, but also with platelets. You can see from the picture that this child has purpura as a consequence of thrombocytopenia. And these patients develop very severe autoimmunity. They are dying from very severe vasculitis, for instance. The protein that is deficient in this disease is that has been named the Wiskott-Aldrich syndrome protein. It's a protein that controls the actin cytoskeleton in blood cells. So a lentival vector has been built actually by colleagues in Italy. You have the name of Luigi Naldini and Alessandro Aiuti who have generated this vector. This is a lentival vector where the cDNA is placed under the control of the promoter. And they did a study in Italy, which has been quite successful, with a follow-up now that extends for 9 years, with limitations for the platelets, as I will show here. That's our own study, where it has been shown in 9 patients that, with a follow-up that now extends to 15 years, that the immunodeficiency is very well controlled. Autoimmunity is limited, but the platelet counts shown on the upper panel is okay, is normal in some patients, but not in others. And this is a function of the number of transduced cells. So we still have to improve a little bit to have a full correction of such a disease. So altogether, and I will stop with this slide, there is further extension of gene therapy targeting stem cells of the bone marrow, targeting, as I just said, inborn errors of immunity like SCIDs, Wiskott-Aldrich syndrome, other diseases like CGD, which stands for chronic granulomatous disease, but other diseases unrelated to the immune system, but where macrophages are used to provide enzymes to control very severe leukodystrophies like adrenoleukodystrophy, a study we did with colleagues in Paris, or metachromatic leukodystrophy, a devastating disease of the neurological system, where that has been very successfully treated now by colleagues in Italy, as well as the MPS 1H, so the mucopolysaccharidose Hurler disease. So these are real advances in the gene therapy today. And to finish with, there is the obvious evolution to also utilize this technology to treat hemoglobin disorders such as beta thalassemia and sickle cell disease, where there are advances ongoing these days. So thank you for your attention.